And all right, so here we are. This is a new way to do the Neuralace podcast. I'm here with Ka Elliott Mathewson, and we're going to talk about a little bit about FNIRS, just a little bit, but it's mostly the topic is about optical imaging. And so let's just dive right in. Kyle, do you want to share your computer? Well, usually I like to start with maybe a short introduction about yourself, just the essential.
Sure.
Go ahead. Anything you want to talk about.
Yeah, while I'm doing it, maybe you can enable screen sharing because I'm
yeah.
Hi, yeah, I'm an associate professor at the University of Alberta up in northern Canada. I got my PhD and did a postdoc at the University of Illinois in Champaign, Illinois, at the Beckman Institute for Advanced Science, where I learned how to record from human brains in a number of ways, including electrically, but also by shining light into the brain and measuring the resulting light coming out the other side. And I run a lab here at the University of Alberta called the Attention, Perception, and Performance Lab. And we study humans out in their natural habitat and how their brains give rise to the behaviors that we do every day. And I guess I could probably share my screen now. Yeah, there we go.
Awesome.
Okay, so, and you'd imagine it's still recording this too, probably, right?
I hope so. We can test this, but
yeah, I think it would.
Okay.
Okay, but you can see this that I'm sharing.
Yes, I can see what you're sharing.
Okay, before, do you have any other questions before I start? Or?
No, go ahead and start. But would it be all right if I do talk during?
Yeah, please. Let's have more of a conversation if we can.
Okay, go ahead.
Well, yeah. So there's many ways to try to measure the activity of the brain, and one of the oldest, of course, is electricity. So you can measure the changes in electrical potential that neurons create as they depolarize and hyperpolarize, either adjacent to them or on them or in them or nearby. But it happens to be, and you can also measure changes in blood flow in different ways, maybe with magnetic resonance imaging or with ultrasound. And because it turns out that as neurons are active, they recruit blood to an area. So even though it happens a couple seconds after neural activity, you can use localized blood flow in the brain to estimate which areas were active in a given situation.
And how detailed can you get? Can you get down to the single neuron? In terms of...
Right. So with fMRI, which is sticking someone in a big 3.5 Tesla magnet and measuring and adding a little bit of electrical energy to shake the protons out of alignment and measuring how long it takes them to be realigned, you can kind of get down to the millimeter or couple millimeter level, which still has probably in each square or cubic millimeter of the brain is 10,000 or more neurons. So you're measuring the human brain at quite a coarse scale using these techniques.
Right. So is that the same for FNIRS? You're talking about fMRI.
Yeah, so with FNIRS, you'll see today, I guess, that at the limit, as we get more and more light sources and detectors, you can approach that kind of resolution in non-invasively in a human.
Okay, that's interesting. So we're talking about like when a single neuron fires, it expands and...
Oh, sorry, yeah, I don't mean you can't get down to the resolution of a neuron. You can get down to the resolution of about a square or cubic millimeter, so a group of 10,000 neurons.
Okay, so we're not yet at the single neuron stage with FNIRS.
Right, at least non-invasively. So you could try by exposing the cortex of an animal or a human in a surgery, you could use some other techniques to do a little better, but the non-invasively or something that could be imagined in the consumer realm we're kind of at this level of groups of neurons. So that being said, 10,000 neurons is a very small subset of like the 10 billion neurons that there are. So these are localized groups that generally are doing the same thing.
Well, you're deep into this niche of optical imaging, and so I'm sure you've heard of all the other optical imaging projects out there, the big names and stuff that promise to do 1 billion times improvement in resolution and actually capture. Do you have like innate skepticism about some of the promises of some of these more headline grabbing?
I would say that there's likely some physicists that understand the limits of some of these technologies, and I wouldn't be surprised if they can be improved on further, and especially with new technologies and expanding on the current technologies. And so I'll talk a little bit about like different types of light that you could shine into the head, and what this new time of flight, it's not new, but what the time of flight optical imaging that Kernel is using, how that differs from the techniques that I use in my lab, or the FNIRS techniques that are more common in consumer devices right now. Let's do that now. So yeah, like let's at least go one slide further. So as I said, when neurons receive messages from other neurons nearby, they receive those messages by neurotransmitter chemicals binding to the synapses, and channels or gates opening up, and ions rushing into the postsynaptic cell. And when those ions rush in, two things happen that we can measure with light. One, we've already mentioned, is that the neuron uses energy to create some of those gradients over which those ions flow. And so to recuperate from that use of energy, it needs to recruit more ingredients from the blood, glucose and oxygen, to make more ATP to power the pumps and gates that let things in and out. And so there's signaling cascades that happen when a local area is active that ultimately lead astrocytes to open up blood vessels in an area and attract, through kind of plumbing forces, more blood to that part of the brain. And that blood absorbs light. And so at the very coarsest scale, you could think of it like a little localized shadow. And so parts of your brain that are more active are absorbing more light, even from the lights in the room and the sun around you. And then a little bit less light is coming back out your head. And then it turns out something else interesting happens when those ions rush into the cell. Water flows in with them because of osmosis, and the membrane of the cell expands and gets stretched out. And it becomes less wrinkly, and it apparently becomes more transparent, and therefore it interacts with light differently. And so then in theory, you can use light to measure both of these things: how much light is traveling through tissue and how fast the light travels through tissue. So the basic idea, well, the first couple studies that were actually done were done in exposed cortex of an animal. So you could actually just open up an image of the brain, a little window into the brain, and take images of that exposed cortex, and you could see it change in color as people use that part of the brain more or less. And I believe there's even reports from Wilder Penfield in Montreal in the 30s of observing change in redness of exposed cortex in the same way.
So I was interested to there's this doctor who's been talking about you've heard of the Medcram doctor, the guy on YouTube
No.
He's been talking about, well, so he's a surgeon, and he's been treating patients with the coronavirus, and he's been talking about how the coronavirus attacks the it targets the ACE2 inhibitors, which are, there's a lot of them on the endothelium, and that's causing basically the damage to the endothelium, and the von Neumann's factor is coming out, and that's creating blood clots, and it's causing vascular constriction, because usually the ACE2 protectors through the angiotensin 2 and angiotensin 1,7 pathway would be inhibiting the vasoconstriction, actually doing vasodilation if you have more ACE2 compared to ACE. And so he's been exploring this, and one of the things they've been doing for coronavirus patients is saying, well, hey, we are sending out we're actually recommending that people have an oxygen detector. And so I was thinking, well, if someone was a pioneering, neurohacker, that they could build an oxygen detector from FNIRS.
Yeah, let me grab mine right now. Yeah, so this is a common technology at the hospital, a pulse oximeter.
Okay.
And it uses the same technique in that maybe we can't even see it, it's hidden in here so well. It has little light sources inside this cavity that shine through your finger, and then it has light detectors on the other end of here.
Yeah.
So this is actually called transmission optical imaging. So the light's transmitting through my tissue as opposed to going in and coming out, which is diffusive optical imaging. But yes, so you can, if you have two wavelengths of light and you're shining them into anywhere into a human body, and they are the right wavelengths, by knowing how much light you're shining in and measuring the amount of resultant light coming out, you can estimate the oxygenation of the blood. Because hemoglobin has 4 spots for oxygen to bind to it, and oxygen coming from your lungs is all 4 spots are taken, and then your body uses 1 or 2 of the oxygen, so you end up with 3 or 2 left on the hemoglobin. And the oxygenated versus deoxygenated hemoglobin absorbs light differentially. Let's see if I have that chart here. Yeah. So tissue with all 4 oxygens in the hemoglobin versus with just 2 or 3 has a different absorption spectra. So here's wavelength of light and absorption. And if you zoom in right around 800 nanometer light, near infrared light, you see that there's this crossing over of the 2 absorption spectra. And so the basic technique of of pulse oximetry, or oximetry of human tissue, is to use 1 wavelength of light over here and 1 wavelength of light over here. I don't know if you can see my mouse.
Yeah, I can see your mouse.
And to estimate how much of the light that you put in is absorbed of those 2 wavelengths, and then you use an equation like this, which is called the Beer-Lambert equation.
Okay.
And you have to know these absorption coefficients. So there, some of the parameters of this equation is the points of your light on this graph, and then what you get out is the change in, or is it, the oxygenated and deoxygenated blood.
Okay.
And really cool. So yeah, now there's work. So in theory, then you can measure that over the whole head, so that yes, as we get the coronavirus, for instance, we might have more trouble getting oxygen into our bodies, but localized across the head, some regions of the brain are using more oxygen than others, and you can estimate those changes over time.
So would you be able to detect blood clots? Because that's one of the things with the, go ahead.
No, keep asking.
So one of the things they were saying with the Medcram folks. So if you go on YouTube and just type in MEDCRAM, the MD who presents it, his name is Roger Seheult, and his lectures are really worth watching for what is the best research I can find on the coronavirus right now. but so one of the things he's saying is that the let's see if I can fast forward. Maybe I can get the chart up for myself real quick. But one of the things he's saying is that let me just go by memory then, is that oops, I lost my monitor. What happened? Oh, wow. Uh-oh. All the monitors are resetting. I guess there was a power outage in the building. Nope, it's back. It's the power, what do you call it, a brownout or something real quick. Let me see if I can go and just say, okay, so what happened was he was saying that there's going back to the ACE2 receptors are basically, they have.
Hey, now I can hear you.
So apparently, like my whole electricity over here got reset, but
Wow.
yeah. So
what a great thing to happen in the middle of a podcast.
We're still recording somehow.
Yeah, I think it's pretty. It's that was kind of the goal of Zoom, was to make it robust.
Yeah. So I don't know, how long was I gone for? I can just continue from.
The last thing I heard you say was the doctor's the leading doctor, and he had this proposal that.
Oh yeah, so his name is Dr. Seheult on Medcram, and he's saying that basically what it comes down to is that if you do not have enough ACE2 receptors, then your body is going to produce too much oxidation or superoxide. I mean, another that's coupled with inflammation or hyperinflammation. So one of the ideas was, well, there are certain things you can get which claim to help reduce inflammation, like on the consumer side of things. There's a supplement called D-ribose. A lot of people try to say that what is these magnesium salts that you soak your feet in, that reduces your inflammation and reduces your so if you could take, people say, well, take your vitamin C and your vitamin D and your vitamin E, and that will help your body to reduce its inflammation. And some people, there's obviously there's ARBs or there's statins, there's different drugs that are meant for people who have heart attacks or strokes that reduce inflammation and reduce they actually increase the amount of ACE2 receptor action. This is outlined in the recent Medcram talks. He does a better outline, much better outline than I'm doing. But the idea is if somehow D-ribose as a fundamental building block of cells is actually, it helps your cells to produce more ATP. If you have this there, the same idea was with the taking the vitamin B12 supplement, which help your body make more D-ribose, which would help your body make more ATP. And so there was this whole energy drink sort of craze where people were taking high doses of vitamin B12. And so the idea is that if any of these supplements are causing an effect where they're reducing your inflammation, there's a point that you could measure. I'm coming back to FNIRS and optical imaging. There's a point where you could measure, I guess you were saying that if someone is having a blood clot, that is going to create a different sort of signal that you're going to pick up on. But if someone is receiving a treatment of, say, D-ribose, and it's reducing their oxidation, and it's allowing their whatever their blood is able to transport in terms of, because the purpose of your blood bringing oxygen is so that you can make ATP, correct? And if you're not able to get oxygen because your ACE2 receptors have been whacked out, but you're receiving D-ribose and you're able to make ATP through other pathways, then the dependence on oxygen. But what I'm curious is that at some point in this chain, would we be able to measure someone getting their ATP from an injection of D-ribose, or it's not a, you know.
Yeah, so there's two cool things. One is that you could measure, well, you could imagine that we're like in the hospital, they're measuring these effects systemically throughout the whole body, but then some of the effects might be more localized to one brain region or another. And so having kind of like distributed sensors around the body might change things. I like your idea though about measuring kind of effects of treatment. There's one cool thing you can do recently shown with optical imaging is you can estimate the pulse wave velocity. So how fast the pulses of blood are moving through the body. And that is an estimate of your vascular elasticity, or how kind of hardened your arteries are. And it turns out you can measure that across the head. So you can measure in localized regions of someone's head, is some of their vasculature more healthy than others. And so that's one maybe kind of thing that could change with some of these treatments, is that your vascular system is working more effectively. But there's a really cool kind of rabbit hole that you reminded me of, and that's that have you ever seen some of these therapies, maybe if you hurt your knee, where they're shining near infrared light into your knee?
No.
Well, it's maybe I can just use the internet instead of my slides here.
Yeah, you can, totally.
Let's try like a near infrared therapy, because there's pretty funny helmets and stuff. Yeah, so this you could get like a belt or something to wear around your waist or put on your knee. And the one idea would be that the heat helps your body, but it turns out that there is in the pathway that creates ATP, there is there's the Krebs cycle, and then there's another 4-stage process that's name is escaping me right now. The electron transport chain. And one of the steps of that electron transport chain is uses a molecule cytochrome oxidase, and it actually absorbs, I think it's something about that step, the near infrared light can make it work more effectively. So in theory, near infrared light can boost the production of ATP locally in a brain area or in a body area. And so I think there's also helmets that you can get that do this kind of thing. Yeah, this lady. Okay, so you can, this guy is treating Parkinson's disease with a bucket that's full of near infrared lights, which doesn't look like a real product in Silicon Valley right now, but I'm sure there's some that are being made like that one or this one. And it's the same idea that you could boost the vascular activity, you could boost the production of energy for the same input molecules. And so maybe your idea that maybe if your oxygen input to the body is diminished, maybe you could acutely improve the activity of those people's bodies so that it's making use of less oxygen to do what it needs to do. That's kind of cool.
Yeah.
Oh no, I think I just closed my slides, but yeah, so I think that's really cool for me. And we've been testing a couple things with this. So we've been testing if you could locally enhance one side of the head and not the other. So you shine near infrared light in one hemisphere and not the other hemisphere. And can you look for changes in cognition that might result from that?
All right.
Okay, where were we? Why are we shining this light in, and how are we shining it? So this is a gentleman named Britton Chance, who was the first to use near infrared, to use kind of non-invasive optical imaging in humans here on himself, had a single light source and a single detector, and he was measuring the amount of light that goes through this path between the source and the detector. And when you use 2 wavelengths of light, as I mentioned, you can estimate the oxygenation of the tissue underneath. And then to kind of test the technique, some studies were done to look for known distributions in the brain, what you would expect to happen. Say if you illuminate the whole right side of visual space, you'd expect there to be activity in the left visual hemisphere. And so you see that in this kind of test of that technique. So you see this boost of activity in the left visual cortex when you stimulate the right visual field, and vice versa. So you can use it as a way to map brain activity, which it's been used for a while. And this is pretty old. So this is maybe 10 or 12 years old, some miniaturizations of the technology. And you can tell how recently I put together some of these slides, because in that time since, there's technologies like this on the horizon that are built into the glasses.
Right.
So this is a company in Toronto that's just in the up and coming stages and is trying to put some of these techniques into eyewear.
Yeah, I've got one of those devices here in my office.
Cool.
Yeah, not the glasses, just the near infrared part. I got one of the developer kits.
Right. And
it's cool. It works with my phone, and I'm working on developing a WebXR biofeedback application with it.
Nice. How do you affix it to your head, or you put it under your hat, or?
It comes with a headband, and then you put it right on your temple.
Yeah.
That's where it's where they recommend you put it. Yeah, I mean, obviously you don't want hair in the way, but I mean.
Yeah, hair is an issue, and that's an issue if you look at the Kernel Hello Humanity post. The kind of the first thing I looked at was the thing I think of the most is like the interface between the human and the machine, and it looks like this is what they're imagining. These are little kind of plastic fiber optics at the end of an optode. Looks like a detector and 6 sources that are about 1 centimeter away from each other. And I'd say that, yeah, this is would have an issue getting in and around hair. So maybe they're imagining it will be on a part of the body with less hair.
Or they'll just, ask their participants to shave their heads or something.
Sure. I'll show you some examples of that too, if we have time.
Sure. Yeah, we've got time.
Yeah.
While you're doing that, I'm going to look to see if I can find my FNIRS device.
Cool. So the basic idea of kind of delivering light into a tissue and measuring the light coming out, you could imagine how these miniaturized electronics could do that job. You have small LED sources, and you have kind of miniaturized sensors that you could embed or kind of attach to circuitry and power quite easily. And
so this is one of the original developer kits.
Wow, cool.
That actually became those glasses. And so you can see it just attaches via headband.
Yeah.
And then I guess you put it on backwards like this, and then it just sits on your temple right there. So
that's great. And does it give you heart rate as well, or?
I believe it does. It's been a while since I've plugged it in, but it's I don't remember seeing heart rate, but it does give you it says right there in the dialogue, it gives you heart rate. I have to check the latest application.
Yeah, well, maybe the heart rate's a good example of how the technique's working, because it was something that took me maybe a year to think about. When we talk about blood arriving at the brain,
yeah,
when you use a particular brain area, what we actually mean is pulses of blood every time the heart beats, and it's in those pulses that a little more blood comes to an area, but it's not like the tide rising in an area. It's that bigger waves of blood arrive at that area. And so inherently in this signal that you get from light over time that goes through our tissue, there's always a heartbeat embedded in it, because every single time our heart beats, it absorbs a little bit more light, that tissue.
What I do like about these devices is that they talk to your phone. I've got an Android phone here to talk to it, and it outputs from your phone. It outputs as a, what is it called? A web socket. So
Cool.
that's good. The VR application that I helped develop in 2018 at Noisebridge, we brought EEG into WebVR. We used a Brainduino EEG device.
Yeah.
And what happened was we created a local server. We used Python, and then later we upgraded it to a server that we created in Go that had some extra features. We added the Fourier transform, and what we did was we created a WebSocket, and then we sent the data out via the WebSocket, and then we brought it into the web page with a WebSocket.
Yeah.
And at that point, we could just use, our JavaScript and our 3JS and our A-frame and our WebXR to take the incoming stream of numbers, divide it up into our delta, beta, our right channel betas, our left channel betas, the whole EEG spectrum. And we could define, what our alpha was exactly, which was nice. But then we could, at the point that it's in a web page, you can say, well, I want these objects, these planes or these spheres or these squares or these cubes to go up and down, and the height is based upon the incoming number that's being received at one interval of time.
Yeah.
And so the blocks go up and down, and we had them in an array. I have video of this. We had them in an array so that they would go up and down in a time series, one for each of the Fourier transform channels, and then they would change colors. They went up and down too. So you could add any sort of JavaScript animation at that point, because you're just making the numbers more reflective. But that's a far cry from making this basically that's a kind of biofeedback application. And if you can get the noise out as much as possible and have a real signal there, then people might find that to be useful. But when you're in VR, you move around so much, and so I thought, well, the next biofeedback application, maybe we'll get a better signal to noise ratio with this FNIRS versus the EEG. But then I'm listening to, I was organizing the Neurotech SF for 2 years, and I'm so glad that my friend Morgan, the amazing neuroscientist Morgan Huff, is now doing that organization, because it's a lot of work. But when I saw, you're talking about doing medical image, doing optical imaging, medical imaging with FNIRS, that's a different kind of computer program that we get the signal, okay, and how could I, and instead of just applying JavaScript variables to each incoming number, well, how would I create a program that actually begins to maybe map out an image, and I don't know how to describe that 3D image. Is it like photogrammetry? Is it like an object file? Is it like is it like in Unity?
Yeah, because that's what kind of drew me to this optical imaging, that I'd already been recording the electrical activity of the brain, and the skull smears the electrical activity. So a good analogy, I think that one of your last guests gave maybe, was that it's like recording, or it was you that gave recording on the outside of a sports stadium, trying to figure out what's going on inside the stadium.
Yeah.
And so that's like what EEG is. And so the idea that instead you could get at least a fuzzy image of what's going on actually inside the brain is appealing. And especially from like a feedback perspective as well, that it gives people not just like a state over time, but kind of an object to imagine. One thing you could try, I guess, with the Blueberry as an easy first pass would be to allow people to visualize their heart beating.
Okay.
maybe with the kind of how dark their setting is or something, because that's a really strong signal, as you'll see probably in this slides, of from the head when you shine light into it.
I mean, there's this with EEG. You know a ton about EEG because you were doing that first, right?
Mm-hmm.
So you know all about source localization, and that's something that I've heard Morgan talk a lot about. One of my questions to Morgan was, why can't we, you know, use basically, you know, if you have a cap to 256 electrodes on it, and you're doing source localization so that you know, basically you can predict where that brainwave may have come from within the 3D volume of the human brain.
Yeah.
Then, you know, why can't you at that point build like, you know, sort of like accurate 3D models of brain activity? And
You can do okay, but I think that the problem is ill-constrained. There's not enough data from the outside of the head to differentiate from two or many possible sources of activity inside. But the best people in the area now are making really, which is the same thing we do for optical imaging, is they're using the subject-specific anatomy from an MRI scan, and they're delineating different structures in that object and how those different structures would transmit electrical signal. And so they're using like really complicated models of the electrical conduction of the head to try to estimate sources, and it takes two different modalities. It takes the structural MRI and the EEG.
yeah. So have you been because I've also been talking to Morgan a lot about doing the structural imaging with electrical impedance tomography. It's something that he's excited about. He mentioned it is helping to solve the skull problem, or I guess the way I think of that is like the how, you know, different people have different skull shapes, and the way that the ions travel through the different parts of the skull is going to be, you know, different from patient to patient. It's also going to be different if the patient's lying down because their brain sits in the skull differently.
Yeah.
And so go ahead and on the topic of EIT if you want, and other options for.
Yeah, I haven't looked in too much to this EIT thing that I hear that Morgan is excited about. There was one thing I remembered of using kind of light, near infrared light, to get a rough estimate of the structure of the brain as well.
Okay.
Because of different absorption or scattering properties in different areas. Yeah, I guess the electrical activity you can measure from the brain, the advantage of it is that it's almost instantaneous in time. You're measuring things as they happen because of the speed with which the electricity conducts. So at least for my research, we try to design experiments that are answering like a when question instead of a where question, because that's what the modality is best suited to answer. Like, how much earlier did it happen in this condition than that condition? But I think that optical imaging can allow you to kind of merge those questions if you also want more ideas about where in the brain some signals are coming from, because it does have better spatial resolution.
All right, let's keep going on optical imaging, because that was sort of like the.
Oh yeah, whatever we can. But I guess I was here and we were talking about how simple it is now and why there's a bunch of companies moving into the space. Because if all you have to do is shine a light or a laser into tissue and measure the light coming out, then, you know, I could take my camera from my phone here, the flashlight from my phone, and turn it on, and I could put it up to the webcam here. And if I just then recorded my webcam over time, or your viewers can pull this video and they can pull my heartbeat from how red my finger is over time.
Interesting.
And so you could put that on your ear, in your head. And so it's any continuous illumination and any light detector. And so that's why Blueberry is able to do what they're doing here. And we were even thinking of making kind of epidermal electronics that adhere directly to the skin, because the technology's advanced that you can make tiny little flexible LEDs and tiny little flexible photodetectors in these kind of circuits. So we were testing out some things like this. And so I think that one of the limitations, and I think that the companies in the space know, is the rigid form of consumer electronics. We're moving farther and farther away from rigid electronics, but nonetheless, there's always a circuit board or plastic involved that usually make things not adhere perfectly to the skin. So one of the areas where I collaborate with people in material science is in kind of imagining what the next form factors of these interfaces between our body and electronics are going to look like.
I wonder if you could measure specifically vasodilation or a single vein, or a map of blood veins.
Yeah, there are. So here's an example. I've seen some other evidence that was like this. So this is like, we put 4 of these lights on a forehead. Let me see if I have a picture. Yeah, so we stuck 4 of these on a forehead. And then using a camera from a phone, sorry, my slides are kind of going haywire. Using a camera from a phone, we just take the frames of that video and pull out the average value of red or green in the frames. And just from the frames of that video, you can get these heart rate out, just as I showed you with that little demonstration on my finger. But usually, and you can see in some images online, you can see shadows and bright spots from veins as well. In the hospital, they have, what is it? There is this technology that nurses use to find veins.
When you said phone, you provoked an interesting thought for me, because one of the basically, I was watching a lot of videos about the Openwater technology that Mary Lou Jepson's working on, and
Yep.
and I came to the conclusion that it seems like the major contribution being promised is a new CMOS chip, or a new phone imaging chip, where it's kind of like you brought up, well, if you just use a phone, you could do some basic medical imaging.
Yep.
But if you had a really good camera chip, like better than the best camera chips available,
yeah, yeah, we're getting close to that. You're right, because we're gonna move past just measuring how much light, and we're gonna start talking about how fast the light moves through the tissue.
Mm-hmm.
And then better cameras are gonna be good because you could measure at a higher frame rate. You're right, trying to also see like how high of a resolution of camera can we use to try to reconstruct underlying tissue. We were thinking about breast tumors or tumors as well, and maybe your idea about blood clots is related to that if you're now moving almost to non-contact physiology sensing, if you can shine lights in from across a room and have cameras nearby, it's not far off from having kind of like a wireless imaging thing that shines lasers at your face and measures a couple things about your physiology. And there's non-contact temperature sensing already happening all over the world today. And there's for sure non-contact heart rate sensing as well. And you've probably seen some demonstrations from Chinese CCTV of them doing that out on the street. I've seen a picture of a policeman with one built into his helmet. Because all you would need to do is pull out the color of someone's face over time, and then pull out the heart rate from that, and then estimate the frequency of that rhythm.
Nice. And if you had a good enough camera, maybe you could get to the point where you're doing, holography,
Sure, yeah.
high frame rate and color in different color changes. Wow.
Yes, and even if the source of light is non-apparent to the person across the room, maybe you have like an infrared source of light that's coming out of your helmet. And just like my neighbor's security cameras have, or our security cameras have, then you don't need to have anything attached to the participant, and you might be able to estimate some local changes in their blood oxygenation in different parts of their body.
And then perhaps you could detect if they're suffering from vasoconstriction and estimate their chances of having the coronavirus remotely.
Yes, that I think the idea of doing heart rate is to do that, but you're right that yeah, maybe you can put this blood oxygenation. Let's check mine right now. Seems to be something that before negative symptoms are occurring, there's changes in blood oxygenation. So I have a 98% blood oxygenation right now.
You did that with your FNIRS device?
Yeah, this little finger one.
Oh yeah, okay.
So I think there's a big promise, and you can see there's people in the space like Blueberry of using continuous light to measure from human physiology, whether it's brains or we already have it in our watches. So on the back of many of your watches is going to be a little light source and a detector that shines through your wrist and measures your heart rate, not far off from measuring your blood oxygenation as well, and it should probably also be able soon to measure your blood pressure. That's apparently a holy grail in these wearable wrist devices, is blood pressure monitoring.
Yeah, well, I mean, if you can detect vasoconstriction or vasodilation, that's the same.
Yep, exactly. So I think that probably what's stopping Apple right now is just a database of known blood pressure values that they can use to train some models.
That sounds like just some lab work. It doesn't sound like it.
Yeah, just waiting for enough people to fill in the survey on their phones or something. Yeah, so so even in this, we were able to measure changes in people's cognitive effort, or at least what looked like changes in cognitive effort when they're doing math versus just calm and meditating. There's less light coming through their head to our phone. And so you could imagine maybe even being able to estimate that regionally across the forehead. And it's hard to know even what product to develop is if you can do that with just a phone from across the room with a laser. But maybe something at airports or shopping malls. So as I mentioned, there's another change in the tissue that happens when neurons become active, and that's that the group of neurons swell up with fluid and become more transparent to light. And so you have a group of neurons that's sitting there and they're not relatively very active, and then they become hyperpolarized and they fire a lot of action potentials, and throughout this time water's rushing into their membranes and swelling them up like grapes. And so the photons of light, which in this diagram are not oscillating, but you could imagine that they're vibrating through the air here, go into the tissue and they bounce randomly off things and scatter through this tissue. So our brain is like the same scattering coefficient as skim milk. So it's like very cloudy. That's why I can't see your brain.
This part in your presentation has really caught my eye the last time I saw it was that it seems like if you had a CMOS sensor powerful enough to this is how you would detect that a neuron had fired, because with this type of tech with light it's not, you're not measuring the action potential, you're measuring the inrush of water and what was it, calcium ions.
Yeah, so it's you're measuring the change in the transparency of the membrane that's because of the inrush of water and the filling up of the interior space of the cell. And you're right that as you see an action potential in that cell, you can see a change in the transparency of the membrane, either in a single cell or this is the axon of a squid, or in like a slice of hippocampus in a dish. So these are classic papers you can see from the previous millennium, showing changes coincident in time with the electrical activity that you can measure with light. And So the basic engineering problem is that you need to measure how fast it takes, or basically you need to measure how much further on average the light goes into the tissue, or you can measure how long it takes to get back to a detector, because it travels at the same speed. So if it goes a little bit of an extra distance, then it's going to take a little bit of an extra time to get back to that detector. So the very simplest thing, of course, you could do is like put in a single photon and then measure at the detector for that photon and pick up the photon and then time it. And it's going to take some crazy fast amount of time because it goes 3 or 4 or 5 centimeters through the tissue, and it travels at whatever it is, 300,000 kilometers an hour. And what you find is in tissue that's active versus inactive, there's a delay in active tissue of like 3 or 4 trillionths of a second in the time it takes light to get through that tissue. So that's the engineering problem, is how do you measure changes in the speed of light that are at the order of trillionths of a second. And the other problem from like a data perspective is that signal big enough compared to the noise that you would get in that system that you build that it's meaningful for your goals?
Okay, fair enough.
So I've shown one way that people have done this, or I talked about briefly, but here's an example of it. This is called Intrinsic Optical Imaging, and it's done in animal models where you open up a window into the brain and you measure with a very high frame rate camera changes in the light that come from an illumination source into the brain and then kind of bounce off the surface and back to your camera. And there's really cool images that come out of this research showing, for instance, on this top right, these are alternating columns of the cortex that respond to the left and the right eye. And so you cover one eye and you show the animal something, cover the other eye and show something, and then you just subtract 2 pictures of their brain. And this is the picture you get in that subtraction. So it's a little darker in some stripes and a little lighter in other stripes.
Okay.
That's from the blood flow, but you can also measure those changes in the time you can measure changes in scattering as well with these techniques. So changes in the transparency of the tissue. And there's work also kind of looking at whisker specific stimulation. So you can see really specific changes in transparency of tissue when you tickle one whisker versus another whisker versus another whisker of a mouse that are coincident in time with changes in electrical activity in that same part of the mouse's somatosensory cortex.
Yeah.
So if you need to measure how fast the light's moving through tissue, there's 2 ways you can do it. So we've already talked back here about what we call continuous optical imaging. So you have a continuous source of light that isn't going up and down in intensity. You shine it in and just measure how much comes out the other side. But if you want to measure how fast light moves through tissue, you can do 2 things. You can use a time of flight device, which is what they're talking about at Kernel, which means that you put in a pulse of photons and you wait at the detector and you measure at the detector how long, but you don't just measure how long, you measure the entire distribution of photons as they arrive at that detector. So because you shine in like 10,000 or 1,000,000 photons in a little fraction of a second, you get a distribution of activation at the detector. And
do you think that they're using a lot of these distributions of activations from different detectors to sort of, combine all of this into a 3D spatial tomography
Yeah, so that's what they're alluding to, and that makes sense because let's go back to this picture here. I think that's a good one. So their idea, the idea of these time of flight techniques is that instead of just measuring the average time that it takes a photon, so the mean of the distribution of the time of photons,
Okay.
if you're shining 1,000,000 photons from here, some of them bounce off and go off into space. Some of them go deep into the brain and get absorbed as heat. Some of them take the path of least resistance, the shortest path, right? And some end up taking the longest path. And so the time that it takes a photon to get the detector is really like a tag of how deep that photon went into the tissue.
Okay.
And so with the technique that I use in my lab, we don't get that measure. We're just measuring the mean of the distribution. So we end up knowing, like, on average, this is how long it took a photon to get from here to here.
I see.
And so we can only assign the measurements that we get at our detector, we can assign them to this whole space. And maybe we might assign them weighted by probability. So like we know that they're most likely to have gone through here and less likely to have gone down here. But what they can do with a time of flight technique, I hope you can start to see now, is they can have kind of really fast channels that go through the surface cortex. So maybe the first quarter of the distribution, they can estimate the median of that time. And then they can have these really long photons, and they can assign the signal that they measure, maybe that's intensity or the speed at which those photons got through the tissue, they can assign to different sections of the depth of the brain.
So do they have to improve the kinds of receivers, light receivers that they have in order to do time of flight on each different portion of the receiver? Is that what's happening, or?
Yeah, so I forget. I know that this is
It comes down to the receiver camera. Like, what is the resolution of the camera in your receiver that's going to enable you to do really precise time of flight and other?
good. Yeah, so this is what is going to be expensive, and this is going to be the hard part. And it's what, like, so the system that we get in my lab does a different engineering trick to measure the speed, and it's maybe a 500,000 or a 250,000 system at the scale that that company would want to produce it. And the current available techniques, I think, of doing time of flight measurements are also quite expensive. So, because you would need to be sampling at a very high resolution. So I think there was some really cool videos.
A high resolution and a high frame rate. I mean,
Right.
that's like one of those cameras. It's like we're it's not just an 8 megapixel camera. It does, like every frame is as good as a photograph.
Oh yeah, so the no, I don't think so, because actually the detectors in these systems are single pixels. So they would usually be a single part of the head that transmits with an optical fiber to a single detector and pulls all the light from that detector. So I think they have resolution in space of 1.
Okay.
And then they spend all their energy trying to record really quickly. But you're right that those demonstrations from MIT, which I think were showing trillionths of a second camera watching light. There's like a video of, yeah, here it is. So this is a video of someone introducing an experiment. Come on, show us the real thing. Yeah, here. So they're recording frames of this video at resolutions of the trillionths of a second. So they're able to slow it down and observe the photons of light moving through this bottle.
Wow.
And so when I showed you our iPhone experiment, where we record light going into someone's head and we record how much comes out, if you wanted to measure the time of flight, like the scattering of the tissue, you would need a camera like this that can measure at that very, very high frame rate.
Okay.
So that's probably one of the advances in these time of flight systems is that they have very fancy cameras.
Nice. What else do you have in your deck? What other things you're talking about?
Let me show you what we do, what our system does. So our system, instead of shooting a pulse of lasers, it is a frequency domain system. So it's kind of the middle ground in cost and the middle ground in benefit. So what it does is it modulates the light going into the head at a megahertz frequency. So it's going up and down in intensity as it goes into the head at, say, 110 megahertz. And that same frequency that's modulating the light is driving a photodetector. And the photodetector, therefore, is kind of expecting light at that frequency. But you also add in a slight different frequency called a heterodyne frequency, which is in the kilohertz range. And then your photodetector can measure at kind of a relatively shorter interval, measure the signal, and it can pull out this oscillation caused by the difference between the detectors and the light source's modulation frequency.
And if you have multiple, if you have a helmet of FNIRs, then you're going to have to oscillate between the different transmissions and receivers so that you're not ruining the image of. Go ahead.
Right. Yeah. So any detector can only know light. It doesn't know where the light's coming from or what frequency it is. So you have to kind of like turn each light on like Christmas lights, or tag them in a certain way such that the detector can separate them.
Okay.
And yeah, so just to show you these frequency domain and the time of flight domain techniques, they can measure slow changes in blood in tissue, but they can also measure these faster changes that happen at fractions of a second in the scattering of tissue.
Okay.
Let me move ahead a little bit here. Right. So the signal that gets measured is you end up with an oscillation because you're measuring the frequency that the light was modulated at, and therefore you can measure how big that oscillation is, and that's this estimate of intensity or how much light went through the tissue. But you can also measure the shift in phase of the oscillation, and that's what we use to estimate scattering, because the photons went into the tissue at one phase of the oscillation, and when they come out nearby, they're at a little bit later phase. And the ones that came through more active tissue will come at a little slightly bit later phase, which translates to these trillionths of a second delay in the photons moving through the tissue.
Nice.
So we can then use that to make these kind of maps like this of neural activity over time. And there's a bunch of boring kind of pre-processing that I've been working on this week of pulling out heartbeats from the signals and filtering them in all sorts of ways. And then also averaging repeated presentations of stimuli to get rid of some of the inherent noise in these signals. The other thing you alluded to was mapping of different spatial areas.
Okay. Yep.
And this is where the consumer electronics are also going to probably start small and scale up towards these kind of research grade techniques where you can record from 30 or 60 light sources that are shined into the brain, and maybe 20 or 30 detectors is the biggest systems out there right now. And so you end up with like 800 or 1,000 different paths of light between sources and detectors. Nice. And if all the sources come on at once, then all the detectors get flooded with light. You get a big image of the brain and you have no idea where anything's coming from. So you end up kind of like Christmas lights, turning different parts of the head on at different times, or modulating light at different frequencies that the detectors can separate after.
So they're showing yellow and red to indicate the approximate distance between, turning lights on from some sensors transmitters to others? Because there's a certain range that you put it in, and so that means you could turn on, looks like a good section of the brain. That's
Right.
as long as the sensors are spaced out far enough.
Yeah. So when light goes into the brain, because of how the brain's like milk, right? If you shine your flashlight into a jug of milk, you'll only see like a bright spot maybe 3 or 4 centimeters away from that light source, because the scattering, just like a foggy day, it makes all these photons dissipate randomly in all directions. And so any light you shine in only goes about I guess it's like 6 centimeters to detectors about 6 centimeters away at the furthest. And so if you shine a light on the left side and you have a detector there and you also have a detector over here, only that one nearby will measure any recognizable photons from that source. So that helps also to measure more areas more quickly, is that you can separate out you can have 4 lights come on at once on different sides of the head.
So one of the things I've been looking for on the side here while listening to you was a question I asked a neuroscientist a while back, and she never got back to me with the answer. But what I asked was regarding hemodynamism in the brain. Is sodium ion movement in neurons attracting blood flow? That was my question. But I think you kind of answered that a little bit.
Yeah, so they're kind of two echoes of, yeah, I wouldn't say so. Sodium transport is a mechanism the brain uses to communicate, and when sodium gets transported, it's because usually a channel opens up through which sodium can pass across an energy gradient that was built up. Yeah. And once it passes through that gradient, now energy needs to be used by the cell to pump it back out to recreate that gradient, because in like 100 milliseconds, it needs to do it all over again. Yeah. And in order to run those pumps, you need ATP.
And in order to get ATP, you need oxygen delivered from the blood, and the other thing was what?
Yeah, and that one's a really weird, and it's more recent that they discovered that like when the neuron starts to pull glucose and oxygen to replenish its ATP, through astrocytes, it signals to the local capillaries in the blood vessels to like let up a little bit, and they get bigger and they allow more blood to flow to that area.
So it's like a pump then. It expands and that sucks in the blood? Is that what?
Yeah.
Okay.
Yeah, so it's not like a vacuum that's like pulling in blood that the astrocyte makes, but it just like your garden hose, it lets a little more area in that space, and that allows more blood to flow to that
area. Okay.
Yeah,
so because I was wondering if there was some sort of, I don't fully understand how the electron transport works, but I was wondering if there's something else that was moving the blood around in the brain, if it's just an area opens up.
Oh, I see. Yeah, no, it is pretty passive. That was something I remember learning as well, that it's not it's not like an active process, but it's passive change in the plumbing that allows more blood to flow to one area versus another.
Okay.
So here you can see we also would use structural images of the people or some atlas to try to recreate exactly which brain areas the light traveled through. And then
one of that, I'm sorry to interrupt you. Sort of going off on a tangent. So one of the ideas that people, a lot of different people are talking about, and I like to talk about this, is the idea that your dendrites can uniquely, your dendrites are little computers, and they can uniquely store, memory information, perhaps in the way they store ions. They might look, a dendrite might look like a tree in its branch-like structure, and it might have little hairs on these branches that store ions in different arrangements, and that might that might have enough sophistication to store unique memory patterns, and then those would be represented in how you're in sort of like the configuration of how your cell is holding, how your nerve cell is holding an electron charge of some gradient of some meaning that's not decoded, but maybe that's where part of the memory is at. And
right, and that's it's kind of in between the way we were thinking about how memory might be stored by like real connections between this neuron and that neuron. It's a little more fragile than that, and then it's kind of the arrangement of the trees of the dendrites.
Yeah.
Yeah, that is interesting. The one good thing to think about is the signal. I'm just going back to this fun picture of the swelling up cell. This is showing the cell body swelling up, but in theory, around the cell body is a huge dendritic tree here. Maybe this is an interneuron, but if this was a pyramidal cell, there's this huge arborization of membrane, that's the dendrites, and there's much more surface area in those dendrites than there is in the cell body. So I show these all as the cell body giving rise to the signal, but it's really the dendritic tree that would change its scattering properties and would be measured by this kind of technique. Okay. For whatever it's worth.
Oh, interesting, yeah.
There must be some more cool stuff here. Yeah, you can see that these contraptions in the lab are pretty homemade still, and they're still kind of honing in on the right form factors of helmet to hold the sensors against the head. They're uncomfortable because they use glass fibers to transmit the light effectively. And they need to be held against the head so that the photons going in don't bounce across the surface of the skin. Hair is an important consideration, and that's another reason to use glass fibers, because they can kind of go in and around the hair. But you can see that for this to become kind of a product one might have in their house, these fibers are going to need to get kind of hidden behind some contraptions or eliminated altogether. Okay. And so some of the headgear development looks like this, that we're using this kind of stuff in the lab now, which is a flexible kind of craft material in 2 layers that is strung together with elastics, so it's rigid against the head, but still can conform to different people's heads.
Right. Yeah, I remember we saw, I believe in, there's a lot of new technology coming out in terms of headgear. So that's one of them. Yeah.
Yeah, there's really cool new stuff of 3D printing specific shapes for people's heads and building the electronics or the optics into those. And I think that's where we're going to head in the lab, because then you can get really good estimation of where you are with respect to their head.
Nice.
This is a boring story that took me a year or 2 of my PhD, but in order to turn those lights, yeah, maybe this is relevant, but if you have 4 different lights coming on different parts of the head and you don't want detectors to confuse them, you need to have them come on far apart from one another such that no detector gets light from 2 of them.
Yeah.
And this is a problem that you can get computers to solve, and that works okay. So you can kind of assign sources and detectors in certain arrangements so that you don't get crosstalk between them. And the final step, which is kind of what you alluded to, I guess, is one of the exciting things about optical imaging is that you could, in theory, recreate an image out of it. And so instead of just getting a signal over time from a part of the head, you could, in theory, recreate the depth of different layers of brain activity or different areas with different sensors nearby one another. You can get better and better spatial resolution because the path of light from each of these source detector pairs are non-overlapping. So every source detector pair has a slightly different part of the brain that it goes through. And the more sources and detectors that you have in an area, the smaller and smaller intersections of them you'll get, and you can estimate better and better localized signal in those tinier and tinier areas. And so there's a couple techniques you can use. You can kind of estimate on average what the path that the light takes through the tissue is. Just assume it's like a banana between the source and the detector, and then assign the data to any brain area in that banana. But then the more complicated techniques would involve kind of modeling the dynamics of photons through tissue, of estimating the optical properties of the skin and the scalp and the pia and the cerebral spinal fluid, and also kind of estimating the differential time that photons that travel deep versus superficially would take. And this is where these new techniques that Kernel is proposing to be developing will be really beneficial, because instead of just this green medium, you could have a gradient in that space, and the fast photons would be assigned to the higher, more superficial areas, and the slow photons to the deeper areas.
So do you have to complete all of this scanning? Because it sounds like a lot of extra work. You got to scan a little bit here and a little bit here and a little bit here. And do you have to complete all of this within a short time window? Because your lymph nodes and your blood vessels and all your, your brain everything moves around, right? So you have to, I would guess there's a time limit.
What do you mean? Or like you mean over the course of an hour, the tissue is going to move around? Oh, I see. Like when you get the structural scan versus when you try to recreate
what's there. Yeah.
Yeah, that's funny, because I have an MRI of myself from 10 years ago that I was just going to use that for now, but you're right that surely my brain has deteriorated considerably in that.
Or grown. Yeah.
Yeah, in some places and not others. Yeah, that's really important. Those are different parts of our university. They're like totally different buildings that you go get the structural scan. So there's actually one process where you take like a 3D representation of where all the sources and detectors were in space on a head, and you take a 3D object of a head and you co-register them together on top of one another, because you collected them at different times. And so you use fiducial landmarks on the nose and the ears that you can identify in both 3D models to co-localize them. And then you use like the shape of the scalp to try to fit the shape of those sensors so that you minimize some error.
So the output of the sensors, are you getting like a point cloud? You're estimating the depth of the points? I'm just trying to picture the 3D data. Does it look like what we're looking at in terms of the 3D?
So both of the two 3D datas are in this plot. One of them is the point cloud of red and orange and yellow dots. And the other one is the grayscale 3D surface. And so you have a point cloud with 3 landmarks, which are shown, I think they're in green on the ears. And you have and there's a landmark on the Bayesian on the nose here too. And you also have the structural image, and in that structural image, you can identify the Bayesian and those 2 points here. So yeah, you have a point cloud and a 3D surface, and you need to overlay them.
But the data you're getting from the sensors is that green data, right?
Not even. No, there's actually no data in this image. The green is the estimated path that the light took when it got to the detector, and then you kind of go through. Is there a good example of that? Sure, maybe here. So the data you get is just numbers over time. It looks like EEG data or weather or stock market data, because any one detector just measures how much the phase of light was delayed over the whole course of the experiment, every 4 milliseconds or 20 milliseconds.
So at this point, you've got depth data, but you're not producing a 3D model. You just have a bunch of 2D models.
Yeah, so for the most part, the depth reconstruction is the least developed aspect of these technologies.
Okay.
And for the most part, we would then project our data up to the surface because, as you can see, the techniques can't image that deep into the brain. They can only measure in kind of the surface area of the cortex anyway. So projecting it up to the surface isn't that.
Have you heard of some open water theory where you could use ultrasound to extend the length of your imaging? I might be wrong.
What do you mean?
So what I heard, and maybe I misunderstood, was that they were able to extend the reach of their imaging by shining the light through an ultrasound frequency.
Cool.
And they're able to get twice as much depth there. I'm sure it's a video that was public. Maybe it's a long now video. I'll send it to you.
Sure, please. Yeah, that's really cool. Yeah, the reason that this depth occurs is because the white matter scatters, like our axons in our brain have fat around them and they scatter the light the most. They're the most like milk. And so once the light hits that white matter, it just kind of bounces really randomly and very little of it arrives back at the detector. So what's really cool about these frequency domain and time of flight optical imaging techniques is you can actually measure like things that are happening at the speed that the brain is working. So one thing you can do is that the brain picks up on rhythmic frequencies out in the world.
Okay.
And so you can flash one frequency on the screen, like 4 or 6 hertz. This is the way some BCIs are used now, that they'll flash different parts of an interactive display at different frequencies, and you can attend or look to some of them. So this is showing how these time of flight techniques could be used for that kind of BCI, I guess, that it can pick out the peak frequencies of the things that you're attending to in the display. And then you can also record multimodal imaging. So you can have EEG sensors built in and around the optical imaging sensors, which I think the Kernel white paper was proposing as well. And so you can use, for instance, like the alpha from the EEG as a regressor on the optical imaging data, which is a little more noisy. Or you could try to look for areas that correlate with like larger, slower changes in EEG activity. And
I believe Kernel was talking about MEG as well.
yeah, so their other imaging technique is using these OPM MEG devices, which are really new right now. And instead of having this large, rigid MEG structure that sits in a room in a hospital, you can have a helmet with kind of MEG sensors that come out of it.
And a Faraday cage to wrap around.
Yeah, and it still needs to be in a magnetically shielded area. So yeah, they're talking about helmets that are Faraday cage helmets. This is a way to combine the EEG and optical. We're working on kind of new software and thinking about this depth reconstruction and how it maps onto existing techniques for visualizing fMRI activity. And yeah, and since this, I've just been working with the MNE Python crews who are working in the same space trying to make tools for FNIRS as well.
Do they have MNE JavaScript? Because I don't know. I'll look it up. Anyway, keep going.
That was it. That was my last slide.
Sweet.
But I can keep talking. Of course.
Yeah, you can keep talking.
MNE JavaScript.
well, we were able to just export anything going on in Python into a WebSocket and bring that into the web page and then apply JavaScript.
Yeah, so that's what I have, this educational website, eegedu.com, that we made this last Christmas
Okay.
break. And one of its functionalities is it can take these streams of EEG data and output them over a socket to other software, or people have used it to control a drone or a robotic vehicle.
Yep.
And the sockets are really cool. Yeah, because you can, we're finding that you can combine sockets. So you can pull in, I'll even stop sharing altogether here. We can start to pull in multiple sockets from multiple users into one place.
Okay. Yeah, well, I mean, there was what was it? There was the software from if you can, what is the guy's name? His Tim Mullen. He has this Intheon company called Intheon, and they make this open narrow pipe software. And I think that what it does is it takes all of your any kind of data signal, whether that's heart rate signal or EEG, and it puts it on a single sheet, a single page, and it's able to synchronize your signals with your it takes your what is it, your time interval, and it uses the time interval to synchronize different kinds of signals. So you could have basically it's not a relational database. It's the kind that allows you to have raw signals that don't match up and synchronize them by time, and then you can make correlations between weird heartbeat patterns and weird EEG patterns and weird eye tracking patterns, and then you can maybe you could use at some point you have all this synchronous measuring of incoming sensor data. Now we're playing with optical imaging and maybe 3D modeling with optical imaging and all these really advanced things. But if then you can correlate that to things that you're looking for. Maybe right now we're looking for is this patient's do they have blood oxygen problems? Do they have are they going to is their von Neumann's factor acting up and causing them to get blood clots? Are we seeing vasoconstriction where in a healthy patient who shouldn't be having vasoconstriction? And are these treatments working? Are these drugs working? What's the impact of these drugs in the vascular system? And these are things that FNIRS could be part of an answer to. But you get this. You have at some point you have. We're gonna see like the low-cost augmented reality device will be like the Oculus Quest. It'll be $400. Maybe it'll have eye tracking. Maybe you'll be able to just clip on your FNIRS device and it'll do blood oxygen, and maybe that's something that people will really want in this new post coronavirus world.
Would you rather know what your stats were or people that you're looking at?
There is definitely room for so there's the doctors who have been talking for a long time about the digital human being, the virtual human being that represents all your vitals, that lets you know about a problem before you get to the hospital. There's a lot of value in that because that's potentially saving your life. So I think that for me to have a digital double, for me to have like a bevy of sensors for my own personal use, it's protected by HIPAA. It's protected by you're doing data collection, but it's behind encryption, homomorphic encryption or whatever, and you.
So I collect a lot of data on myself and my heartbeat and steps and exercise, and I found that my physicians don't have time to consider all my data. Like there's too much data that I can collect that my physiotherapist or my family doctor's like, that's way too much for me to consider right now. I don't want to see your your heart rate 3 times a day for the last year.
Right. But when you have too much data, I'm looking to see if I get my hand in the picture. When you have too much data, I was like, where's my hand? Where's my hand? The thing to do with a lot of data is to apply neural networks, because that's where neural networks thrive, is when you have a lot of data. And if you could get the neural network to basically take a huge amount of data, but summarize the semantic meaning of that data to something simple that a doctor can glance at, and then make a decision to either confirm the prediction or estimate of the AI and turn it into a diagnosis, because the AI will present the most relevant facts after pouring through the mass amount of data. And that's the goal that I want to get to. It's not just having my data collected and presented as a virtual human, but having the AI crunch it to its most salient point.
Because right now they've put the onus on the user to be like, here's your heart rate every moment for the last year, but what do you, like I've graphed it, it changes, but I don't know what to do next.
You don't have a semantic meaning estimate of what that data means. And that's the
my
point.
Family doctor and the healthcare system doesn't yet either. Like they haven't learned what they should keep track of. And
there's so much of medicine, I think, is based on, like, this context. We ask you so many questions to determine if these numbers are relevant to your context. And that's just really messy in terms of, how medicine works. But we could maybe get better answers if we not only collect this data, but have, do a lot of hard work to figure out how to apply neural networks to crunch this down into semantic meaning that's relevant to your life. And that's a huge, like, for me, that's a task somebody could be doing right now.
And who owns the data? Like then it's a data sharing agreement, and the user gets the data and the companies. Like how do we motivate that middle between the data and the knowledge?
There are companies that are working on answers, like Verily, there's a Google company, they're working on, well, how do we do data sharing? How do we profit somehow? Or how do we, make it so that, like, I've heard people, why does blockchain come up in this context? But the idea was, well, we want people to be able to share encrypted data. We want that encrypted data to be validated, but still, like, only people, you own it in a sense that you choose which doctors can see it. It may be out there on the internet on everyone's server, but then only special people have the special glasses that can see what's in that file. Until then, it's like this really expensive public, blockchain data is a lot more expensive than just storing it.
So I guess my concern would be that the way things are going, that the knowledge from that data isn't shared, the value from the knowledge from the data isn't shared with the producers of the data. That's the concern. That if there's some value in my data, it should be to kind of, and you want to motivate me to collect it, it should be beneficial for me. It should give me feedback about my physiology, or when I should see a doctor, or what I should be eating, or what I should be exercising. It shouldn't just be used to make kind of higher level decisions about the healthcare system. But yeah, so how do you have to sell the user on the benefit to them?
And users are not going to, there's too many stories of companies and just regular people discriminating against people. Oh, this person has the coronavirus. Let's go mob their house, and let's like deny them insurance, and they can't have insurance now because they have a pre-existing condition, and all this stuff. You don't want your medical data to be known in theory, because of all the weird irrational ways that people could attempt to discriminate against you. And there's lots of examples of that. So yeah, I don't know. I think you really want to be able to control your data and decide what hospitals get it, but also to control it, because hospitals go out of business. And then what happens to those records? They're lost. Hospitals are going out of business right now, and that's a real problem facing people. Their data is not on some server that they can transfer to another hospital. So, and they got to do all the tests again. But I think we should wrap it up there. This was a great call. Thank you.
Fun.
We learned a lot about optical imaging and 3D imaging, 3D optical imaging, but there's a lot more. There's a lot more to learn, I sense, and so there will be more talks in the future to dive deeper into everything that everybody cares about with mathematical imaging. There's so much more to discover and talk about. And thank you very much, Kyle.
Yeah, thanks,
And
Michael, for having me.
hopefully, I'll go ahead and test the video now, see if it recorded properly.
And we didn't just have a nice chat for an hour. It was good. At least it wasn't. I sometimes record lectures for my class and it doesn't record at all. And I've literally just talked to no one for an hour. So at least I was talking to you.
Okay. All right. Talk to you later.
See you, Michael.